Welcome to Chapter 12, The Cell Cycle. Here is a series of drawings depicting mitosis of a plant cell. Cell division is divided into two major parts, interphase and mitosis. During interphase, the cell is growing and functioning but not actively dividing, but it is preparing to do so by copying genetic material at one point. About 90% of the cycle is made up of interphase. Interphase consists of the G1 phase, the S phase, and the G2 phase. Chromosomes are only duplicated during the S or synthesis phase of interphase. After clearing the G2 phase, the cell will actively divide into the next major part, mitosis. Here is a diagram depicting the cell cycle. The size of each slice is relative to how much time it takes. Mitosis itself consists of 5 major phases: prometaphase, metaphase, anaphase, and telophase. During G2 of interphase, the centrosome divides into 2 centrioles and the spindle fibers begin to form from each one. The first stage of mitosis, prophase, sees the mitotic spindle develop as the sister chromatids begin to condense and thicken. At this point, sister chromatids, that is, copies of each chromosome, are visible. At the next stage, prometaphase, the nuclear envelope disintegrates and microtubules are formed, many of which attach to the middle of each sister chromatid pair on its kinetochore. During the next phase, metaphase, attachment creates movement along the spindle fiber. Each side pulls in a sort of tug-of-war as each pair of sister chromatids lines up near the middle of the cell along the metaphase plate, an imaginary axis line. Anaphase occurs soon after. Anaphase is known as the migratory phase because this is when the sister chromatids separate, being pulled to opposite ends of the cell by the spindle fibers. Once they are separated, the sister chromatids are now individual chromosomes. The last phase, telophase, is when the rest of the cell separates into 2 daughter cells that are genetically identical to the parent cell. A nuclear membrane forms around each new set of chromosomes as they begin to become thin, and the rest of the cell divides in the process known as cytokinesis. Cytokinesis in animal cells involves a cleavage furrow that forms to separate the 2 new cells. In plant cells, cytokinesis causes a cell plate to form between the new cells, eventually laying the groundwork for a new cell wall to be formed. And then we see the cleavage furrow forming in a dividing animal cell during cytokinesis. And in this image, we see a cell plate forming between the 2 new plant cells.